Hi everyone, today we've got the very last part of aerobic cell respiration, the electron transport chain. So everything we've done so far, we're going to start putting it together. So far, where are we? Well, we started off with glycolysis. Glycolysis made a little bit of ATP through substrate level phosphorylation, thanks to those chemical reactions. Glycolysis also made a little bit of NADH. The pyruvate traveled into the mitochondria where it started the Krebs cycle. The Krebs cycle made a little bit of ATP, again, through substrate level phosphorylation, and it made a lot of NADH and FADH2. The NADH and FADH2 electron carriers went to the electron transport chain, and the ETC is going to make a lot of ATP through the process of oxidative phosphorylation. So that is what we're going to learn about today. So again, the point is to make lots of ATP. So far, glycolysis has 2 ATP and Krebs has 2, but life is going to need much more than 4. Some very simple organisms might be okay with 2 or 4 ATP per glucose, but not something as complicated as a large eukaryote like a human. So we have all kinds of working cells that need lots and lots of ATP. So there has to be a better way. And there is a better way. The electron transport chain evolved, which is a great way for our bodies to get tons and tons of ATP. The electron transport chain is a series of proteins built into the inner mitochondrial membrane. So that is where this takes place. So that is along what I referred to yesterday as the cristae. I'll probably only refer to it as the inner membrane. Transport proteins and enzymes are going to be necessary for this to happen. And we are going to be transporting some electrons down this chain, which is going to help actively transport or pump some hydrogen ions, which are going to create that gradient that we've talked about before. This is going to yield much more ATP, about 36 just from one tiny little glucose, and that is more than enough to keep us alive. This can only happen in the presence of oxygen. So sorry, anaerobic organisms cannot undergo this process. They undergo a different process called fermentation. This very efficient process can only happen in the presence of oxygen. Okay, so here this is going to happen on the highly folded inner membrane. This process is really important, and that is why the inner membrane is so folded, because we need as much surface area as we can for the electron transport chain to happen many, many times. So it's happening on this high surface area, highly folded inner membrane. So that's what we're going to focus on. So here is what the electron transport chain looks like. This is the inner membrane. These blobs are some proteins. This is the intermembrane space. So this is the outer membrane up here. The matrix is down below. These are the names of the enzymes. You don't have to memorize them, but you'll probably become familiar with NADH dehydrogenase, cytochrome B, cytochrome C. So another few little proteins in there. So this is what it looks like, right? These 3 proteins that are going to act as membranes are very important. So we have all of these electron carriers. Glycolysis gave us 2. The Krebs cycle gave us 10. So we have 12 per glucose. We have 10 NADHs total and 2 FADH2s total. That's quite a bit of energy carrying electrons. So we have the stored energy in the form of electrons that we are going to now cash in. We're going to break open the piggy bank and take what's inside and make it usable. So NADH breaks down into NAD plus and hydrogen when we release an electron. The hydrogen is going to break down into an electron and a proton, a hydrogen ion and an electron. So that happened here. NADH meets up with NADH dehydrogenase, this first protein, and it drops off its electron and it loses an H. The H is here, And the electron gets pulled through the protein to this next part. This next part is more electronegative than the first part. So the electron is attracted and the electron is pulled down through this protein. So as the electron is pulled, it loses energy and it allows a hydrogen ion to be pumped through. So you should imagine this protein undergoing a conformational change. As the electron is pulled through, that conformational change that's happening pumps the hydrogen ion across. Okay? The electron continues to get pulled down because now this second protein is even more electronegative than this protein. So the electron continues to move. It's getting pulled thanks to electronegativity, and it's pumping more hydrogen along the way. And then the electron continues to get pulled thanks to electronegativity. And once again, we're going to pump even more hydrogen. So the intermembrane space up here is filling up with hydrogen ions. FADH2 has to bind at a different place. FADH2 is going to bind here. And when it gives up its hydrogen and electrons, it is going to do so on this second protein, pumping hydrogen as the electron is pulled down. Eventually, the electrons meet up with oxygen. Oxygen is found at the very end. There's an enzyme here which will hold oxygen, and that will attract the electron finally. And oxygen will meet up with some hydrogens that are floating around to make water. So what we're doing here is we are building a proton gradient. So the question here, what powers the proton pumps? So think about that. What is powering the proton pumps? It is the movement of the electrons. So the electron movement is really important. So stripping hydrogen from the electron carriers allows the electrons to pass down the ETC. So the hydrogen is cleaved off, the electrons are stripped, and the electrons are passed from one protein to the next. They go down the chain, getting pulled all the way down. The flowing electrons, the electrons are losing energy as they move. That energy is transferred to the proteins, which can then pump the hydrogen across. Pumping there, and pumping there. The transport proteins in the membrane are going to pump hydrogen across the inner membrane into the intermembrane space where they are being built up. And that's going to give us our gradient that's going to cause our ATP synthase to work. So what pulls the electrons down the electron transport chain? Oxygen. We have to have oxygen to make this work. Oxygen is what is basically going to be found at the end of the ETC. It's going to be right here, and that is what pulls the electrons down, and then it reacts with the electrons to make water. If there was no oxygen found at the end, the electron transport chain would stop, and nothing would happen. There would be no pumping, there would be no gradient, and therefore there would be no ATP production. So electrons flow downhill, and the final electron acceptor is oxygen. I would write that down. The final electron acceptor is oxygen. And that is why this is called oxidative phosphorylation as opposed to substrate level phosphorylation. So the electrons flow downhill to oxygen, so they lose little bits of energy as they move. And that energy is going to be captured by those proteins, which can pump the hydrogen across. It is a controlled oxidation, and it's a controlled release of energy. And that is how we still obey the laws of thermodynamics. The energy of the electrons goes down as that energy is transferred to the proteins. Instead of making a fire, like in our example of burning wood, we are making ATP. And we finally did it. We set up our hydrogen ion gradient, which allows the protons to flow through the ATP synthase which is then going to synthesize ATP. So ATP is going to get made. The gradient allows the hydrogen ions to move through. The movement of the hydrogen powers the synthase to make ATP. That's a proton motive force. A term you should know is chemiosmosis. Chemiosmosis is the diffusion of ions across a membrane. So this builds up a proton gradient and then allows for the diffusion of those hydrogen ions through the ATP synthase. That is chemiosmosis, the osmosis of chemicals or ions. So chemiosmosis links the ETC to the ATP synthase. And that's the point. This was discovered by Peter Mitchell. This model was proposed by him in the 60s. He won the Nobel Prize. So to review, electrons are harvested, they go to the ETC. The electrons are moved, that provides the energy to pump the protons into the intermembrane space. The oxygen joins with some of those protons and the electron to form water. And the protons diffuse back down their gradient, synthesizing ATP. Okay, so glycolysis gives us 2 ATP. The Krebs cycle gives us 2 ATP. And the electron transport chain gives us 36 ATP, which is going to net us around, it's not an exact amount, but it's going to net us around 38 to 40 ATP total. All right, so some questions for you to think about. Where did the glucose come from? That came from food. Where did the oxygen come from? That came from the air. Where did the carbon dioxide come from? That came from the glucose. Where did the carbon dioxide go? It went out the body through the lungs. Where's the water coming from? The water came from the oxygen. Where did the ATP come from? So that came from the ADP that was made by the movement of protons. And what else is produced is not listed in this equation. I'll let you think about that right now. And why do we breathe? Oxygen in helps us move the ETC, carbon dioxide out, that gets rid of the waste. What is the final electron acceptor in the electron transport chain? That would be oxygen, very important. So what happens if oxygen is unavailable? The ETC backs up, there's nothing to pull. The NADH and FADH2 can't unload their H's, and ATP production is going to cease. And the cell will run out of energy, and then living things cannot live. All right, and that is where we are going to stop for today. It's an example we'll talk about later. So our point, to make ATP, and that is just what we did, about 40 ATP. Let me know if you have any questions.